Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. How are you, dear viewers? I hope all of you are well and fine. Dear viewers, today in this video we are going to see another culture media, that is the Ashdown's Agar. Dear viewers, if you remember, we already talked different culture media. The playlist of the culture media will be present into my YouTube channel. You can go back and watch the playlist that is related to the culture media, okay, if you miss the culture media's lectures. Today in this video we are going to see the 37th video lecture that is related to the culture media, and in this video lecture we are going to see another culture media, that is the Ashdown's Agar. So what is actually Ashdown's Agar? Ashdown's Agar, it is actually the selective media which is specially used for the isolation and characterization of a bacteria which we call that Burkholderia. Okay, this is a type of bacteria that can cause milioidosis. It's a type of disease. Now this type of media was first discovered by a scientist named L.R. Ashdown in 1979. That's why the name is Ashdown's Agar. Remember, this type of media is specially used for the isolation of Burkholderia specimen that we can taken from the non-sterile sites like sputum. Then we have the compositions. So this type of media is composed of different ingredients, like we have Tryptone soy broth 10 gram, we have agar 15 gram, glycerol 40 ml, gentamicin 4 milligram, crystal violet 0.1%, 5 ml, we have neutral red 1%, that is 5 ml, and we have distilled water 950 ml. Okay, so these are the ingredients that we need while making this type of media. Now we have the requirements. So for the requirements, we need culture media, petri dishes, autoclave, incubator, personal protective equipments. In personal protective equipments we need goggle, gloves, overall, cap, etc. Okay, we need glassware like beaker, flask, etc. We need wire loops, spirit lamps. So all these things we need while processing this type of culture media. Now let's see the principle. The first thing that we are the first thing that is mentioned in the culture media, that is the crystal violet and gentamicin. As all of you better know that the crystal violet and gentamicin are the ingredient that are specially responsible to suppress the growth of different type of the bacteria. So we have only the selective bacteria can be easily grow. Okay, now we have the colony of pseudo Burkholderia pseudo meli. So this type of the bacteria using the neutral red, and when it is using the neutral red which is present into the media, what will happen? It will be distinguished from the other type of the bacteria. Then we have gentamicin. So gentamicin is a specially type of the antibiotic that can inhibit the growth of what? It can inhibit the growth of Burkholderia pseudo meli, and it can also allow due to this way different types of other we can say bacteria can be easily grow of the Burkholderia species. So whenever we want to grow the bacteria on this type of media, so we need 48 to 96 hours, okay, incubation time. Then we have glycerol. So the main purpose of the glycerol is it specially required for the this Burkholderia pseudo meli. So then we have 24-hour incubation. So remember, when we place the plate into the incubator for 24 hours, so what will happen? The colonies are often pinpoint, clear and pale pink. Okay, and it can be changed pinkish, purple, flat and slightly dry after 2 days incubation. Then we have the characteristic feature of Burkholderia pseudo meli. It is actually metallic sheen colony, and it usually progress to dry and wrinkled colonies after 96-hour incubation. This is the principle. Now let's see the preparation. It's very simple. First you need to mix all the ingredient the measurement which I told in the previous slide. Okay, so you have to measure all the ingredient, then you will mix it into 950 ml distilled water, and you will heat to mix boil all the ingredient gently. Then you will autoclave it. Autoclave process will be 15 minutes at 121 degrees Celsius. The pressure will be 15 psi. Okay, now after the autoclave, then you will wait for the media, so the media will cool down at 50 degrees Celsius. Then you will pour it into the sterilized pretty plate. Then you will wait for the media to be solidify. Okay, now what will you do once the media get solidify? So then you will streak the bacteria. After that you will examine the bacteria culture, okay, onto the media, and you have to examine different type of Burkholderia culture after every days within the weeks. Okay, now this is the result that we get after incubation. So these are the bacterial colonies, and these are all the species of the Burkholderia. Okay, so this is all about the Burkholderia selective media, we can also say that, and this is all about the Ashdown culture media. I hope this video is helpful for you. Thank you so much for watching.